All right, so at this point we've talked quite a bit about lipids, and we've probably covered the more typical waxes and fats that you probably know a fair deal about already. So we'll turn our attention to another kind of lipid in this lecture. So specifically, we'll discuss phospholipids today, and phospholipids, of course, are something I would imagine you've heard of as a group, but maybe haven't looked at the chemistry of in detail. So we'll take a little while here and look at some of the different types of phospholipids and get a better sense of their chemistry. The first type of phospholipid we'll discuss are the glycerophospholipids, and these are the most abundant lipids in cell membranes. They're similar to the fats we've described, at least the triacylglycerols, in that they contain two fatty acid residues, so they've made these ester bonds with the first and second hydroxyl group of that glycerol, but in that third position we formed an ester bond with a phosphoric acid that then forms another phosphoester bond with an amino alcohol. And we haven't talked about amines yet. We'll be looking at amines in the next chapter, but we'll obviously take some time here to discuss amino alcohols with respect to the glycerophospholipids in this chapter. Specifically, lecithin and cephalin are glycerophospholipids that are abundant in brain and nerve tissues. They're also found in egg yolk, wheat germ, and yeast, and they contain choline, which is in the lecithins, and either ethanolamine or serine in the cephalins. So hopefully you get a fair deal of these through your diet, but certainly soy lecithins are often additives to other foods. So even if you're not eating egg yolks or wheat germ or yeast, you may be taking in some lecithins as additives to other foods. If you read those labels, you'll be surprised what you see sometimes. Okay, so as promised, we're going to talk a little bit about those amino alcohols. Three amino alcohols that are found in glycerophospholipids are choline, which we mentioned is found in the lecithins, and then serine and ethanolamine, which are found in the cephalins. So in the body, at a physiological pH of about 7.4, these amino alcohols are ionized. So if you look at the nitrogen, which is the amino group, as we'll discuss in more detail next chapter, the nitrogens have a formal positive charge on them for the choline, serine, and ethanolamine amino alcohols. The other end, the OH at the other end of the molecule, has no charge, right? It would be very unusual to have a charge on that group. You'd have to be at extreme high pH and again, not physiological conditions like we have here. But to protonate the amine, to protonate that nitrogen, is fairly common at that physiological pH. So there we have it. When we talk about amino alcohols, we mean that they have an amino, or actually an ammonium group here being charged, and then an alcohol group. And so we've dealt with alcohols already, but the amino alcohols, the amino group, we'll talk about in more detail next chapter. If you're really anxious to learn more about those, then you please move ahead to that chapter as soon as possible. Now, the glycerophospholipids are interesting because they're both polar and nonpolar. That's not unusual in this chapter on lipids. We've already seen that sort of behavior, but in particular, we have these polar and nonpolar regions that allow interaction with both polar and nonpolar substances. So if we're talking biochemistry, there's lots of water around, right? So that ionized amino alcohol and the phosphate portion, which we call the head, those are polar head regions, and they're strongly attracted to water. The hydrocarbon chain known as the tail is soluble in nonpolar substances and would interact well with other hydrocarbon chains in a nonpolar type of interaction. So we've got both the affinity for water at that ionized amino alcohol phosphate region, and then we've got a fear of water, hydrophobic region there at the hydrocarbon chain. So not anything unusual for our discussion of lipids, but certainly features that allow us to understand why these would be common in cell membranes where we have that lipid bilayer. So we already mentioned it briefly, but let's talk about that formation of glycerophospholipids again. So it happens through ester linkages, of course, more traditional ester linkages for those two fatty acid residues, and then what we call a phosphoester bond between that phosphate group and the glycerol that contains those two fatty acids, and then of course the amino alcohol, which in the case of lecithin it would be choline, and then in the case of acetylene we might have something like ethanolamine or serine. So here in this image we see those residues. For the fatty acids we just have something like palmitic acid represented here, but of course they can be different. They don't have to be the same. Just for simplicity, they're shown as the same fatty acid residues here, but of course what makes them unique and different is that the final linkage there, the phosphoester bond that has the phosphoric acid group and then the ionized amino alcohol. So that's, as you see, the polar region is around that fully charged formal charge of +1 on the nitrogen end of the amino alcohol, and that gives rise to a lot of affinity for water along that whole group. The phosphate group as well, having a full negative charge there, is also going to interact well with water, and then the fatty acid groups up above will not. They'll interact well with each other and with other fatty acid residues, but not with water. So here we see another representation with those general components. So we show the fatty acid residues here. Now they're different residues. We have a saturated residue in the first position, then an unsaturated residue in the second position. And because of that kink, we have free rotation, of course, so we just switch the side on which we show that third residue, which is of course a phosphoric acid that forms a phosphoester linkage to the glycerol, and then another phosphoester linkage to, in this case, choline. So this must be a lecithin that we formed, and they're particularly unpleasant smelling if you have a pure sample of them, but when they're present in small amounts in food, it's not noticeable. And of course they are an important and necessary part of our biological chemistry. So there you see it, the typical components and how you might form those ester linkages by taking those alcohol and carboxylic acid groups from the glycerol and the fatty acids for those first two residues, and then making those phosphoester bonds by that phosphoric acid residue and the glycerol alcohol, and then the alcohol from the amino alcohol. Now notice in this image we've made those bonds, we've made those ester or phosphoester linkages, and so we have the glycerophospholipid formed, and we can see clearly that we have that polar head group on one side of this representation of the molecule, and then we have the nonpolar tails. So if we were going to make a lipid bilayer, then we'd have the ability to have these polar head groups aligned along the inside and outside of the cell membrane, and then in the middle region there of the membrane, you could have those nonpolar tails interacting with other nonpolar tails from the other part of the bilayer. Okay, so let's pause here to have a learning check. In this particular learning check, you're asked to identify each as a fatty acid, triacylglycerol, amino alcohol, or glycerophospholipid. So we've got some names there. They may be specific names, they may be generic types, but hopefully at this point in the chapter, the names at least seem familiar, and you'll be able to identify A, B, C, and D as one of those options up above. Stop the video here while you make those assignments, and start us back up when you're ready to check your work. Good luck. Okay, so hopefully these are what you assigned for the different types. So A, glyceryl triolate is a triacylglycerol, right? It's glycerol forming ester linkages with 3 oleic acid residues, and that's its name after it's formed that triacylglycerol. B, cephalin, that's a class of glycerophospholipids, so hopefully you were able to make that determination. Choline is a specific amino alcohol that we mentioned is present in the other type of glycerophospholipid, not the cephalins, but the lecithins. And then finally, palmitic acid is a fatty acid residue, 16 carbon saturated fatty acid residue. So hopefully you were able to get all those correct. If you happen to miss one or two, hopefully now you understand why this is the correct assignment of types. And if that's not the case, if you're still struggling, do a little extra reading, try another practice problem or two, and then certainly reach out to me for help if it's still not making sense. Okay, let's follow up with one more learning check. In this one, we're asked to identify the components and type of glycerophospholipid present. So hopefully those residues look familiar, probably more so for those first two, which are identical residues. Uh, the last one there with the phosphoester linkages may be a little more difficult to identify because we've just talked about them in this part of the chapter 17 lectures, but please stop the video here, try to identify each of those components, and then the major class of glycerophospholipid present. Once you've made those assignments, start us back up and we'll check your work. Good luck. Okay, so hopefully you had a little trouble identifying those as stearic acid residues. That's probably the most common fatty acid that we've discussed so far. It's 18 carbons, all saturated. That's stearic acid. Here it's connected by an ether linkage to a glycerol in positions 1 and 2. Then we have in position 3 that phosphate forming that phosphoester with the alcohol group that was formerly on that glycerol. And then that's serine, our amino alcohol here. So because it's serine, we know we must have a cephalin. Serine or ethanolamine would be our amino alcohols for acetylene. We talked about the lecithins would have a different amino alcohol. So here, because we have serine, we know we must be acetylene as opposed to a lecithin. You've got to love the names of these amino alcohols, right? We had choline for the lecithins, we had serine or ethanolamine for the cephalins. Now we've got sphingosine, which is an 18 carbon unsaturated amino alcohol. And so you see it there. It has that carbon-carbon double bond there, just one carbon after the carbon containing the hydroxyl group, or at least one of the hydroxyl groups, the hydroxyl group at the top left anyway in this representation. Then we've got another hydroxyl group down a couple carbons later. So the other ones that we've talked about, the ethanolamine and the serine and the choline, those were all saturated amino alcohols. This time we have an unsaturated amino alcohol, and the sphingosine, as you'll see in the next slide, is going to be a pretty important building block for some of our other lipid types. I pointed out in the previous slide that we had multiple OH groups, and that's important for sphingosine because the sphingolipids are a class of phospholipid that contain long-chain amino alcohols called sphingosine, which we learned about previous slide, rather than glycerol. So instead of those triglycerides that we've come to know and love, whether we're talking about the triacylglycerols from a previous lecture or the lecithins or cephalins from this lecture, the phospholipids, they were all based on that glycerol backbone. And in this case, the sphingolipids are based on sphingosine instead. And you can see where there's a couple functionalities there like glycerol, but certainly some differences as well. Now going even further within the sphingolipids, we get the sphingomyelins, which have that amine group forming an amide bond to a fatty acid. Again, amines and amides, that's our bread and butter for the next chapter, so don't worry if you're struggling with those ideas just now. Certainly when you get to the next chapter, you'll have those concepts under control before you go ahead and take the next exam. Notice also that the hydroxyl group of the sphingosine forms an ester bond with a phosphate to form another phosphoester bond to an amino alcohol. So similar to what we saw for the phospholipids, the cephalins and lecithin. And then in this case, we'd have another amino alcohol such as choline. So sphingosine is an amino alcohol, and then we'd have another amino alcohol like choline present in the sphingomyelin class of molecules. Now, if you know your biological chemistry, you probably noticed that myelin name in the sphingomyelins, and they're abundant in that white matter of the myelin sheath, the coating around the nerve cells. So they're also abundant in cells that increase and speed the impulse, those nerve impulses, and insulate and protect the nerve cells, right? That's really what that myelin sheath is all about, and it's an important function, a necessary function. Certainly without these sphingomyelins, nerve cells can't carry those impulses correctly or at all, depending on whether we're talking about nerve damages and things like that. So the sphingomyelins are quite important, and they have quite a very important function there in nerve cells. Just to underscore how important those sphingomyelins are, let's take a look at multiple sclerosis, which is a disease, a disorder in which the sphingomyelins are lost from that myelin sheath, and therefore the neurons in the brain and spinal cord don't have that same insulation and protection. Scars then form on those neurons, impair the transmission of nerve signals, and if you've ever seen or know anyone who suffers from multiple sclerosis, it's a very difficult, debilitating disease, and it's a result of these sphingomyelins not being present in the amounts necessary to perform that function of coating and protecting the nerve transmission in nerve cells. So on that note, we'll go ahead and end this lecture. We won't end with a learning check as we normally do, but hopefully this real-life application, I know many of you are interested in the health professions, so hopefully this link here is a good enough way to tie up this chapter on phospholipids. Okay, we'll see you in the next section of chapter 17.